Hello, everyone. Thank you very much for joining us today for our webinar on light sheet microscopy, titled Advances in Light Sheet Microscopy, the Universal Tool for 3D Bioimaging. My name is Malte Wachsmuth. I'm the Head of Applications, Support and Service at Laxendo. We are part of the Fluorescence Microscopy business unit of the Bruker Nano Group. And in the next couple of minutes, I would like to walk you through the concept of light sheet microscopy and give you an overview of our product portfolio, with which we try to cover a very broad range of applications. Who are we? Laxendo, we are the light sheet fluorescence microscopy brand of Bruker's Fluorescence Microscopy Group. We started as a spin-out from EMBL in Heidelberg, Germany in 2015. Completely dedicated to light sheet microscopy. What we're doing is we're developing technology really around the application. And we joined Bruker, the Bruker Fluorescence Microscopy business unit, in May 2017. Before I move on, I would like to mention that at the end of the presentation, there's time for a couple of questions. And as you may see on the right side in the control panel, there is a questions panel that you could then use to put questions. Okay, let's get started. What we want to do in biology and in bioimaging, we want to image basically biological objects that cover a quite broad range of orders of magnitude, starting on the molecular level in the nanometer range, and the largest objects we might want to look at are animals on the size of 10 meters, like a whale. What you can look at with the naked eye is everything above, let's say, the thickness of a hair, which is 100 microns, 1/10 of a millimeter, and up to whole animals. That's our normal world we're living in. And if you want to image smaller objects, cells, bacteria, viruses, we usually use light microscopy that covers the range from a few hundred nanometers up to a couple of millimeters. And if we want to study objects below that scale, viruses, proteins, and individual molecules, and we typically use electron microscopy or X-ray scattering methods, for example, crystallography, to get an idea about the structure. As you know, over the last 10, 20 years, light microscopy has undergone very dynamic development towards resolution enhancement, but also towards imaging of larger objects based on the development of modern objective lenses, of a lot of advancements on camera technology. Laser technology has evolved a lot, and all this helped us a lot to develop microscopes that can image further to smaller objects and to an improved resolution. Our general challenges in light microscopy are that we want to image large volumes. At the same time, we want to image this at as high resolution as possible. And we want to be very fast with our acquisition speed to capture, for example, the beating heart of a medaka fish. And at the same time, we want to be gentle enough so that we can image for hours and for days objects in 3 dimensions with very high resolution without affecting the biology. And all these somewhat, to some extent, contradictory requirements are something that we would like to combine, and we want to expand on all these axes of microscopy as much as possible. And light sheet microscopy is a tool that helps a lot to really cover all of these 4 axes and to improve in all directions. The evolution of light sheet microscopy started more than a century ago. The first light sheet microscope was conceived by Zsigmondy and Siedentopf in 1903, where they first demonstrated the combination of an orthogonal arrangement of an illumination optics together with a detection microscope. Then this concept got forgotten for like almost a century, until 1993, when Roy et al. Published a paper on light sheet imaging of the inner ear cochlea of a guinea pig. This was kind of like a one-time event until 10 years later, in 2004, Ernst Stelzer and his group published their landmark paper introducing what they then termed selective plane illumination microscopy, or SPIM, for live cell and live sample imaging. That's the concept, and they applied this in the beginning to zebrafish and to Drosophila, so the application range was more in the range of developmental biology. And since then, LSFM, as it is also abbreviated, has really undergone a very dynamic development until, for example, in 2014, Nature Methods, the journal Nature Methods termed this the Nature Method of the Year. And the concept of light sheet microscopy is really very simple and very straightforward. The idea is that you basically decouple the excitation of a fluorescence signal in your sample from the detection beam path, like this is highlighted here in the sketch, where you have the light of a laser coupled into an illumination optics that is focused with a cylindrical lens in one direction, whereas it remains unaffected in the other direction, which results in a sheet of light that is then directly illuminating a sample in the sample chamber that can be filled with an appropriate sample medium. And this light sheet then happens to be in the focal plane of a second optical path, which is this detection path that you see here, which is a quite conventional wide field detection scheme with fluorescence filters, a tube lens, and then a high-end camera like an EMCCD or an SCMOS camera. That's the idea, and this provides a lot of advantages compared to confocal imaging, for example. On the right side, you can see a photograph of a light sheet microscope that we built at some point in our lab at EMBL in Heidelberg, where it basically illuminates your sample with one objective lens to form a very thin light sheet here, which is then the focal plane of an identical objective lens here to the right that images the fluorescence excited in this focal plane, in this case, the fluorescence solution, to highlight the concept on an EMCCD camera. What's the advantage compared to confocal imaging or fast spinning disk confocal imaging? What you do in a confocal microscope, you basically scan your object point by point, line by line, frame by frame to acquire the fluorescence distribution in 2D and in 3D. This is a sequential imaging process because you're illuminating just a single spot and you're collecting the light excited in this focal spot, and using a pinhole, you're rejecting a lot of light that might come from out of focus planes. What you do at the same time is you're illuminating your object hundreds and thousands of times also outside the focal plane, so the whole volume basically, without collecting information from that part of the sample, which results in a lot of unwanted fluorescence, unwanted illumination, and thus photobleaching and phototoxicity. Also, the sequential nature of this approach makes this imaging concept reasonably slow. And if you want to be very fast to capture fast processes, there are a lot of limitations there. In order to overcome the latter limitation, the idea came up to parallelize this process by using, for example, spinning disk confocal systems. They make use of a disk containing a lot of little pinholes or lenses that focus not a single spot, but hundreds and thousands of spots in the sample. And then you follow the same scheme. You collect the fluorescence from all these illumination spots to generate a 2D and a 3D view of our sample. This makes the whole process much faster, but you still have the limitation that you're illuminating large fractions of your sample without collecting information, and the ratio of illuminated volume of your sample to the volume from which you actually collect information is still not very good. And this is something you can overcome using light sheet microscopy. What you do in light sheet, you take a second optical path to illuminate just a single plane of your object, thus the name SPIM. And this then happens to be in the focal plane of a second detection path, where you can then make use of camera-based imaging so that you image all pixels at the same time, which makes the whole imaging process much faster. And at the same time, you're just illuminating that part of the sample from which you collect information. In summary, this dramatically reduces the light dose that you put on your sample, so you have less phototoxicity and less photobleaching. You don't need any pinholes to reject photons. You basically collect everything that comes from the sample. You don't waste photons in a way. And at the same time, you have an optical section capability that is very comparable to that of a confocal microscope. So you're faster, you're far more gentle to the sample, and you get the same 3D information, which makes light sheet microscopy the ideal tool for imaging, on the one hand, dynamic processes in live specimens, but also for fast imaging of very large objects, for example, cleared brains of mice. This is our favorite movie that has been acquired in the lab of Lars Hufnagel at EMBL, one of our co-founders of Luxendo. And the idea here was to be able to image all individual nuclei that are highlighted here with the histone marker in developing Drosophila embryo over like 24 hours. And they wanted to track every individual nucleus over time so that they could not tolerate any photobleaching, so that otherwise they would just lose the signal. And they needed a very high time resolution to not lose track of every individual nucleus. And this was only possible with the light sheet microscope, because then the time resolution at the same time, the level of phototoxicity was low enough so that they could image for 24 hours almost with a 30-second, half-a-minute time resolution. But you cannot only look at Drosophila embryos or zebrafish embryos, you can look at individual cells, you can look at spheroids, at 3D cell culture, at C. elegans. People use light sheet microscopy to image Arabidopsis or even starfish. So the range of biological samples and applications you can cover with light sheet is very broad. And the question is, at the end of the day, how do I build my light sheet microscope so that I can image the sample I'm interested in? When you look at the literature, there's really a very broad zoo of geometries that have been suggested for light sheet microscopy. And the criteria that you might want to use to choose your light sheet is basically, what's the sample size? Well, how can I mount my sample without affecting the biology? And what is the spatial resolution and also the temporal resolution that I need? Resulting in basically all kinds of combinations of illumination and detection objective lenses arranged around the sample so that the sample is happy and develops properly over time, whereas we can image without photobleaching a lot. In the following, I will focus on these 4 geometries because we offer light sheet microscopes covering all these 4 geometries. The first one is a multi-view light sheet microscope with 2 opposing illumination objective lenses and 2 detection lenses. This is a horizontal arrangement, so imagine this being basically the horizontal plane. Another geometry is imaging from below and illuminating from 2 sides so that we minimize shading effects in the sample. A third approach is another inverted light sheet microscope, which you see here in panel E, where we illuminate from one side under 30 to 45 degrees illumination angle, and we detect in another perpendicular direction to collect light from the sample that is then mounted in a quite conventional inverted approach. And alternatively, you could also flip everything around to look at your sample with 2 objective lenses from above for illumination and for detection. And in the following, I will basically explain to you what you might want to use for which application and how we covered this with our technology approach. This is an overview of our product portfolio. So we have all these different geometries. The first one is the movie SPIM, multiple view for live samples, which has then be extended by a photo manipulation module. There is an option to also image cleared samples with this, and we can also combine all these modules together. The second family is what we call the inverted view light sheet microscope in SPIM, where we have an inverted geometry, as already mentioned, which can also be complemented with the photo manipulation module and with an advanced illumination module for more complex illumination patterns. The third kit is the QV-SPIM, which we call quantitative view, which is an upright geometry. And we've very recently expanded our portfolio with 2 inverted geometries, the LCS-SPIM, the large cleared sample light sheet microscope to image really large objects that are typically cleared, and the TrueLife 3D Imager, which is a dual-sided illumination inverted light sheet microscope for larger live objects like spheroids. Our product portfolio is complemented by a powerful acquisition and processing software called Lux Control. We also offer a kind of an intermediate data storage and processing solution to help you to store, process, and archive your data. So what do you use when you want to image entire living organisms like a Drosophila embryo or like a zebrafish? What we suggest to use is then typically the movie SPIM, the multi-view light sheet microscope. And multi-view is for a reason. So when you're imaging an object of a couple of hundred of micrometers in diameter, like this zebrafish embryo that you see here, provided by Antonio Giraldez from Yale, when the light for illumination travels through such an object, you will then notice that after a couple of 10 or a couple of 100 micrometers, then the light is absorbed and scattered and all the signal deteriorates. The same applies to the detection part. So when we combine two illumination directions and two orthogonal detection objectives, then we have 4 combinations of illumination and detection, each of them optimized for 1/4 of our object. And since we combine this and image this simultaneously, we get all these 4 views at the same time without the need for sample rotation. And then by computationally registering and fusing the data, we will end up with an optimized view where we remove the scattering and the absorbing part of the light, both for illumination and detection, to have an optimized view on our sample, which then allows to acquire data like this, which we have acquired at the zebrafish course at MBL in Woods Hole 2 years ago. And what we've acquired here for 18 hours is the zebrafish as it develops in our sample chamber. While it's imaged, we acquired all these 4 views with 2 cameras that I mentioned, and we image every few minutes for like 18 hours without any detectable photobleaching and phototoxicity effects. The system we used for that is the movie SPIM. That's what it looks like. It's effectively a black box containing all the electronics and all the lasers we use, and all the optics is put on top of the microscope. What you see in the center is carrying the objective lenses. We have a 4-lens geometry, 2 detection lenses, 2 either 20x 1.0 MA water lenses from Olympus, or 2 Nikon 16x0.8 NA water lenses, depending on your requirements. We use 2 Hamamatsu Orca flash cameras for imaging, so really state-of-the-art CMOS cameras, and we can image with up to more than 40 frames per second in full frame acquisition, which allows, for example, 22 volumes of 200 planes per minute, so the time resolution of full 3D imaging is quite decent. We can offer up to 6 and now even 7 laser lines simultaneously, ranging from 400 to 780 and 5 nanometers. And we are mounting the sample from below and also offer optional photo manipulation, as I will tell you in a minute. The central part is what we call the octagon, which carries the illumination and the detection objective lenses. We have 2 filter wheels with 10 positions, covering really a broad range of fluorescence detection windows for many, many fluorescent applications. We then have an additional magnification, deep magnification optics to cover a broad range of magnifications as a zoom optics. And then the light goes to the already mentioned Hamamatsu cameras. We're illuminating simultaneously from 2 sides, the sample chamber, and we're also detecting simultaneously in 2 directions. So we don't compromise on time resolution because we do everything at the same time. That is what the sample chamber looks like with a slightly zoomed in. You see the light sheet coming from 2 objective lenses from the top and from the bottom. We do this by arranging the objective lenses such that the 2 light sheets perfectly overlap along the optical axis, but they are slightly displaced axially, so that we have effectively an extended field of view. We are using a Gaussian beam to illuminate the sample, which has a lot of advantages in terms of flexibility, in terms of chromatic corrections, which is then scanned vertically to generate a light sheet in the sample. And the properties of the Gaussian beams are such that when you make the Gaussian beam reasonably thin, then it becomes also quite short, which is then called the Rayleigh length. So if you want to, what's this? Sorry about that. If you want to extend your field of view, then that's why we're using these 2 actually slightly displaced Gaussian beams. This is a close-up of the sample chamber, and what you can see there is the sample as it has to be put in between the objective lenses. And this sample is basically sitting on a post that can take, for example, a glass capillary holding a little object. The sample itself is a 3D translation and rotation stage, so we can really optimally position it between the lenses. And the sample post basically takes up, for example, a glass capillary into which we embed the sample based on low melting agarose. The way it works basically is that we melt low melting agarose of, let's say, 1% concentration. So that we can then put our object, in this case a Drosophila embryo, inside the low-melting agarose as it's still fluid. Then we suck it into a glass capillary, let it solidify inside the capillary, and then we push it out again and then stick it into the sample post. The advantage is that the agarose has the same refractive index as water. So when we embed it in the water bath here, it becomes invisible, but at the same time, it provides a rigid support of the sample. So it doesn't float away, but stays where it is for imaging. Alternatively, you could also use what is called FEP. That is a Teflon material having as well the same refractive index as water. So you could stick your FEP tube, for example, on top of a glass capillary, which then serves as a little mug into which you can embed your sample. Especially helpful for samples that do not like to be embedded in agarose, that where the biology is basically affected by the physical constraints of agarose embedding. This is an overview of field of view, pixel size, and resolution that I don't want to read you through. You can look this up in the recording or on our website, or just get back to us, and we are happy to discuss this in more detail. Just want to mention that with our latest edition of a 2x additional magnification step, we have at a resolution, at a nominal resolution of the objective lens with an NA of 1.0 of about 300 nanometers. We have a pixel size of 140, so we have proper oversampling also for deconvolution concepts. The travel range we offer is for the 20x 1.0 and a higher resolution lens 1.2 by 1.6 millimeters, whereas for the 16x 0.8 NA lens, the travel range is 3 by 3 millimeters. So we can image quite large objects. Now this is also new that we extended the travel range, especially for the 16x lens, and the travel range in the third dimension, which is perpendicular to the image plane here, is in the range of 12 millimeters. So that's basically the object size or the range you can cover with this microscope. Let me show you a couple of examples. This is a zebrafish. This is the beating heart of a zebrafish, imaged by Professor Jing Xia in Huazhong Agricultural University. They are basically interested in cardiac development and deviations in cardiac development. At the same time, you can also zoom out and image the whole fish while it's alive. This is a stitched acquisition with several tiles stitched together, and you can hopefully see that even at such a large field of view of several hundred micrometers up to a millimeter, we can still have cellular and subcellular resolution and also a very good temporal resolution. This is basically the same object imaged in a high-res, high-speed mode and then in an overview mode to image the whole object. That's another example of a mammalian tumor growing on a zebrafish, which is also a 3D acquisition from one of our customers in China. This is a very nice example of nuclear tracking in zebrafish embryo acquired by C.S. Smits from Stan Schwarzman lab at Princeton University, where we have a movie SPIM. And basically the data were then used to track individual nuclei using Aridis Vision 4D. Our data are compatible with all common tools that are available, Aridis Vision, Bitplane, Imaris, and of course all the open source Fiji tools. How do we create a light sheet in our system? And in the movie SPIM, we are creating the light sheet using a Gaussian beam that is then scanned vertically to generate a light sheet over time. This has been published in 2008 by Ernst Stetzer's lab and by Philip Keller, who is now at Geneva Farm, termed the DSLM, the digitally scanned light sheet. The more conventional approach is that you're using a cylindrical lens to generate a static light sheet, which is focused in one direction and unaffecting the other, so that you illuminate the whole sample, the whole imaging plane at the same time. We are using the second approach in most of our systems. We have one more advanced approach that I will come to a little bit later. You can also benefit from this approach when scanning the light sheet. There we make use of a property of SCMOS cameras that you may know from your mobile phone when you acquire a movie of something rotating very quickly, then you see these funny distortions of the objects like a propeller. And the reason for that is that the whole image sensor is not read out at once, but line by line, which is called the rolling shutter functionality. So there's a temporal relationship and a temporal delay between the lines at the top of the image versus the bottom of the image. When you synchronize, however, the scanning process with the rolling shutter functionality, you can define a sensitive area and then have a confocal-like suppression of scattered light, for example, without affecting the time resolution and the spatial resolution. So you get rid of light that you don't want to collect because it's scattered, it comes from out of focus planes, and you basically just record what you're actually interested in. And this is an example of a comparison of a mouse embryo with and without this, what we call the line mode. You can hopefully see that with the line mode, you can get rid of the haze inside the object very much without affecting or even while improving the signal to noise ratio of the information and of the images we're acquiring. If you want, in addition to imaging, if you want to interact with your sample, you can also do this based on our light sheet movie SPIM system by adding a photo manipulation module. You can see this little backpack attached to the right side and the scan optic sitting in here. It's a 2D Galvo-based manipulation approach where light of, for example, a visible laser somewhere in the range of 405 to 780 nanometers is coupled into the high NA objective lens to illuminate individual spots and regions of interest in the focal plane while you're imaging. You can also add pulse 780 or 1040 nanometer lasers for photoablation, wounding, and coagulation, photo-induced coagulation, for example. So this provides a very flexible tool for interacting with your sample while imaging with very high time resolution. Alexis Mezel at the University of Heidelberg and his postdoc Marion Louveau, they provided us with this image here. What they do, basically, they are interested in lateral root development in Arabidopsis, and they deplete individual cells and then observe what the impact of the depletion of these base cells for the lateral root development is. This is another example that has been published quite recently from EMBL in scientific reports. What they do is basically what they did is they put an IR laser to introduce damage here in this area of the neuron, and then they observed the microglia coming and doing their job at this position. These are applications you can actually do with a photomanipulation module attached to our movie SPIM light chain microscope. What if you have different applications, for example, 2D or 3D cell culture, organoids, spheroids? So when you're closer to what is usually imaged in cell biology, then you would probably opt for another geometry. I mentioned already that sample size, sample mounting, and required spatial and temporal resolution define what you want to do. And in the following, I will introduce this microscope based on the geometry shown here in E, which we call the InVSPIM, standing for inverted view light sheet microscopy. The concept and the motivation were such that in the lab of Takashi Hiragi and Jan Ellenberg at EMBL, what they wanted to do is they wanted to image very early mouse embryonic development, really from time point zero. What they could do very easily, they could easily mount their oocytes on a cover glass and image on an inverted confocal. However, after a couple of imaging rounds, the confocal put too much light, and then it basically resulted in a lot of photobleaching, and they couldn't really image with a good time resolution for more than, I don't know, a couple of hours. They wanted to image, however, for 2 days or a couple of days. Then they put their mouse oocytes in a movie SPIM geometry. There, they had to mount it inside the gel, inside the agarose that I mentioned already. And why they could image for a very long time with a very high time resolution, the gel resulted in the freezing of the biological development of the mouse embryos. They just stopped dividing at some point. That is something you just realize after 24 hours of imaging, most likely due to the physical constraints imposed by the gel into which the sample was mounted. So therefore, the idea came up to have something like a curved cover glass, so that you can basically put your object in between your objective lenses, the two that at least that you need for light sheet imaging, and that's basically what the idea that into which this resulted. You have a curved cover glass, which is effectively an FEP foil that can be filled with the correct medium and with your sample, of course. This is then embedded into an immersion chamber that is maintained at the right temperature, for example, 37 degrees. And then an illumination objective lens looks at the sample for illumination, and a second detection lens for the quite conventional imaging path. The whole chamber that you see here, so this concept, the sample is put here into this immersion chamber, and then you can close the lid, and the whole black thing is then like a little incubator that is held at the right temperature, for example, 37 degrees, and you can provide high humidity to avoid evaporation of the medium. You can also provide the right CO2 and the right oxygen concentration for the sample to develop properly and to survive. This allowed then long-term imaging of these mouse oocytes. I will show you a couple of images in a minute. Conceptually, the system is detection-wise a quite conventional one. This is the incubation chamber I mentioned already. We have again two CMOS cameras, in this case for two spectral channels. And then when we illuminate our sample, we detect it with a detection lens, and then we split the two channels by using a dichroic mirror. So this is a quite conventional approach using the same cameras. It's a single view, single illumination, single detection view, which is good enough for very transparent objects like mouse oocytes or smaller 3D or 2D cell culture. When you look at the system from the front, you will then get a better idea of how to mount the samples. Opening the lid, you see the sample holder, which is sitting inside the chamber. And what we use to actually mount the samples is a piece of material called PEEK. PEEK is a plastic material that can be autoclaved and is very inert and biocompatible. We then glue FEP foil into this little chamber so that it sticks out at the bottom to form this little trough here. And this is then the window through which the objective lenses look for light sheet imaging. So you don't need any agar rolls, no glue or something like that to mount your sample. The sample is just dropped into the trough and then held by gravitation inside the sample chamber. Then you close the lid and you have an incubator for imaging of a couple of hours. We also partnered with EBD to create a disposable chamber. I will talk about this in a minute. This is the EBD-based incubation system, environmental control system that we also offer. So it's a gas control unit to provide the right level of humidity, oxygen, and CO2, so that you can also generate hypoxic conditions for embryology applications. And this is a Peltier-based water cooling and heating device, which also allows to cool the sample chamber down to 20 degrees Celsius and below, so that you can also potentially image, for example, marine samples that sometimes don't like warm temperatures very much. This is a summary of all the specifications I mentioned. Let me highlight the lenses we are using. We are using a 0.3 NA 10x objective lens for illumination, and most importantly, a Nikon lens with 25x nominal resolution and an NA of 1.1, so that we collect as much light as possible. It also provides automated access to the correction color, so that we have also means to correct for slight refractive index mismatches, up to 6 or now even 7 laser lines between 400 and 785 nanometers. And it's very straightforward to keep the sample sterile because of the topological separation of the sample from the immersion solution. This is an example of the first paper that were acquired with a prototype of our microscope at EMBL in the lab of Jan Ellenberg, Takashi Hiragi, and Lars Hufnagel. These are pre-implantation mouse oocytes that were imaged over time, and what you see on the right side is the condensation and first intermingling of maternal and paternal DNA. And then at some point you will see the formation of a metaphase plate and then the separation of the sister chromatids. This is basically the first time this has been really imaged over time properly without phototoxic effects. So this was only made possible by light sheet microscopy. On the left side, you see more of the cells over time, and you see the divisions as they happen more or less in a more or less synchronized manner. And the idea here was to basically figure out which cells become outer cell mass and which become part of the embryo, and only through imaging for a long time and then looking backwards through the time series, you can basically tell this and then classify the cells and try to find out what determines their fate, the lineage fate. And this was also only possible through light sheet microscopy, because only with a light sheet microscope you can image long enough and often enough so that you can track cells over a long period of time. This is an example of a cluster of a 3D spheroid provided by Pierre Neveu from EMBL, formed from mouse embryonic stem cells, and you can see this is about 180, 200 microns in diameter. You can see that we have a very good lateral resolution XY, but also the axial resolution is very close to the lateral resolution. That's another advantage of light sheet microscopy. When you make the light sheet very thin, which defines then the Z resolution, you can really obtain a very close to isotropic resolution. Nevertheless, you might see some striping, and there are also other ways, and some deterioration of the signal when the light travels through the sample. So you see illumination is here from the left, and you see that there's some shading going on, and the question is, can we somehow help to remove this? And yes, the answer is yes, there are ways to do this. I will come to this. I would like to mention that also this system can be complemented with a photomanipulation module with the same property. So it couples into the 1.1 NA objective lens to allow depletion of cells, to allow photoablation, but also photoactivation or photobleaching in a more conventional FRAP or optogenetics experiments. So as said, what if the samples are a little bit too big for illumination from one side? And if you don't want to give up, what if these samples are really relevant for your research? And that's a very current example of a paper that came out in May, and a nice comment from Anne Grappin-Botton, who's director at the Max Planck Institute for Molecular Cell Biology and Genetics in Dresden. And she said that organoids are really important, are an important approach to understand how a virus infects which cells in the body. And this is, of course, about the SARS coronavirus 2. And this is a study on how coronavirus infection goes on in three-dimensional vascular and kidney organoids. So towards a better understanding of the actual infection process introduced by the SARS coronavirus and the resulting COVID disease. So how can we do this? The answer is just add another objective lens, which takes us to scheme here in panel D. And our system that allows to follow the scheme is our TrueLight 3D imager that we launched half a year ago. It is again an inverted light sheet microscope, but with dual-sided illumination. So conceptually, it is such that we have our object again in such a sample holder based on FEP foil, but we are now illuminating from two sides instead of one, and we're detecting the fluorescence signal again with a 25x1.1 NA lens from below. This can be operated like in the InViSPIM at about 30x and 60x magnification for either an overview imaging or an high proper oversampling, high resolution imaging. The whole sample is again embedded put into this incubator-like approach. You have this lid again that forms an incubator, and conceptually it's very similar to the InViSPIM, except that it's a 3-lens geometry with dual-sided illumination. Also, what we changed is that we increased the length of the sample chamber to 75 millimeters, so we can image far more samples. This is then optimal for multi-position imaging of embryos, but also 3D spheroids, all sites as already described in the example. And as already said, we teamed up with EBD to create our disposable true life 3D dishes that can be put into the sample holder, but they can also be used on the InVSPIM. So they are compatible with both systems. They come as perfectly sterilized packs of 6. And you can just put them in the sample holder. They come with a little holder so that you can put your samples in there. You can put them in an incubator for a day before your actual experiment. So it's a dish like any other dish you would use for your experiments, except that it's compatible with our light sheet microscopes. So the way it works is when you put your samples, like several hundreds or hundreds of little embryos in this very 75mm sample holder, you basically insert it into the sample chamber, which provides, again, full incubation with CO2 control, oxygen control, humidity, and temperature. And then you can image many of these cells in 3D over time. So you really have the possibility to do highly multiplexed experiments. And when you use, for example, 3 of these EBD true life dishes, then we have 6 separate compartments you can image in 6 different conditions. The whole detection scheme is again the same. We are imaging with 2 spectral channels with Hamamatsu Orca flash cameras. Applications are time-lapse and total imaging of small embryos, as said, 3D cell culture models, spheroids, organoids. That is something for which the system is really very well suited. You can track stem cell development and also look at in vitro fertilization. Just a second. I have an issue here with the sharing. Okay, sorry about that. This is an example of pancreatic spheroids provided by the lab of Anne Grappin from Dresden. We imaged them. Those are pancreatic spheroids derived from human embryonic stem cells, and we use the True Life 3D imager to image not only one of them, but really many of them put into the chambers of our True Life 3D dishes with long-term imaging. The visualization here was done with BitPlane Imaris. So if you don't necessarily need dual-sided illumination, but you want to illuminate your sample with a somewhat more sophisticated approach to, for example, improve resolution, or to use, for example, Bessel beams to increase the imaging depth with what is called then a self-reconstructing beam, we have developed a module that we can add to our InVSPIM that is called the Advanced Illumination Module. Compared to the regular InVSPIM, you see this L-shaped box sitting on top of this, which contains our Advanced Illumination Module that is an SLM-based device that allows to generate more complex pattern beyond just a regular Gaussian beam. So we can generate, of course, a static Gaussian beam. We can generate a scanned Gaussian beam. But we can also generate a Bessel beam that we can scan across a field of view, or we can generate parallel sets of Bessel or Gaussian beams, which is often referred to as optical lattices that can be scanned or dithered across a sample for faster and low phototoxicity imaging. The question is then, what's your application? Do you need more high speed? Then you would probably go for a static Gaussian beam. If you need a good compromise between speed, gentleness, and resolution, you might want to go for a Gaussian beam. If field of view is key to your application, you really have to make use of your whole 200 or 400 micrometer field of view, then a scanned Bessel beam might be the best choice. And if you are really focused on resolution, then using an optical probably the best choice for your application. And the good thing with our system is that you don't have to realign anything. You don't have to do much. But if you want to switch between the different imaging modalities, you just click a button in the user interface, and then you can switch from the Gaussian beam to an optical lattice. And you can very pragmatically play around with what is the best solution for your application. So the specs of the InVSPIM Lattice Pro are very comparable to that of the InVSPIM. Plus, we have this SLM technology for creating the different illumination beam profiles, including your own, if you really want to design your own illumination profile. And we're using a higher MA 0.7 MA illumination objective. Everything else is very similar. Just a word on the resolution. When we measure the resolution with a dithered lattice, we end up with a lateral resolution of 219 nanometers, which one would expect using 100 nanometer tetraspec beads. That's basically the physical limit of the objective lens, and the axial resolution is then less than a factor of 2 larger. It's in the range of 470 nanometers, also pretty close to the physical limit of what is technically feasible with this approach. When we do this with a more conventional Gaussian beam, we have the same lateral resolution, but in significantly higher, twofold higher axial resolution. So this is really close to what one would expect. And this shows us basically that it's very easy to switch between different modalities and yet have an optimized configuration for each and calibration of the system for each imaging pattern. As said, you can also use this in a very pragmatic way to get an idea or just decide what's the best solution for your application without having to spend too much time reading all the papers on lattice light sheet microscopy. That's an example of HeLa cells expressing labeled tubulin from provided by Sabine Reiter from EMBL in Heidelberg. When you use a Gaussian beam, you see that the Gaussian beam is best for the central part of the field of view. Then you have a decent resolution and also quite good signal to noise ratio. When you then swap to a Bessel beam, the resolution and the signal to noise ratio remains the same here in the central part, but the whole usable area of the field of view is really expanded to the whole 210 by 210 micrometers in this example, which means a Bessel beam provides the same resolution as a Gaussian beam, but just a larger field of view. The Bessel beam, as you maybe know, has a lot of side lobes, so this results in a bit more photo bleaching. But if you can tolerate this, if you don't need long-term imaging, then this is probably the best choice, because then you can cover the whole field of view. As comparison, we also use an optical lattice, and in this example, this didn't work very well. So at the end, you basically decide, okay, Bessel beam is the best across the whole field of view in terms of resolution and contrast. In another approach, again, very pragmatic, we tried this with 3D cell culture. These are spheroids labeled with EGFP and MRFP. And it turns out that when we imaged the two channels sequentially, for the green channel, the optical lattice gave the best results, whereas for the red channel, the Gaussian beam gave the best results. This is a sample provided by Martin Stöckel and Elisa May from Constance University. So at the end, you can very pragmatically combine the Gaussian beam in one channel and the optical lattice to get the best multicolor view of your sample. And this is an example of the same cells that have been used for Eric Betzig's paper, just to provide you also with an idea about the time resolution that you can get. A sample kindly provided by Hartmann Hartz from Munich University. That's another example of epithelial cell lines stained for F-actin and chromatin, provided by Ulrike Engel from the Nikon Imaging Center here in Heidelberg. And you can hopefully see that we can really get a very nice resolution of our sample across a quite large field of view. This is a spectrally split view of all these different channels that were provided here. And another example with imaging over time, making use of the incubation functionality in the system, are these HeLa cells expressing histone 2B fusion protein and GFP tubulin. This has been visualized using Imaris, and this hopefully shows you that you can nicely image with high resolution over longer periods of time. So let's move on to another geometry that we can offer. This is an upright geometry that is in our overview scheme here, panel F. So instead of looking from below, we can also look at the sample from above, which has a lot of advantages in terms of fields and in terms of sample size that we can image. And that is our system. For this, we offer the QV-SPIM, the Quantitative View Light Sheet Microscope, which is in a nutshell an upright microscope with two identical objective lenses, which can then look into single chamber SPS plates or customized sample chambers that are optimized for your application. We have two orthogonal views that can be used to then generate isotropic view. I will explain in a minute how this works. We have full environmental control, temperature, CO2, oxygen, the same as for the InViSPIM. And we offer either two Nikon 40x.8 NA lenses for water immersion, live imaging, or two ASI special optics 16x.5 NA lenses for multi-immersion imaging that can be used for live imaging, but also for imaging of larger cleared objects. Again, we offer up to two simultaneous spectral channels. And this is basically the concept behind this. We have our two objective lenses that we use sequentially, one for illumination, one for detection, and they just swap the roles, and then we obtain two orthogonal views of our sample. What does it mean in terms of data processing? We get this view A with a view with illumination from the left and detection to the right, and then we swap and we have the illumination from the right and detection to the left, and these two views overlap, and with computer-based registration and fusion of the data, we can generate a nice isotropic 3D view of our sample. As said, we have these two spectral channels that we can use for imaging. So with our two lenses, swapping the roles, we can image then two spectral channels on two Hamamatsu cameras again. That would be path one, and that's path two for the imaging. That's an example with two views that you can see here. The Bauer lab at MSK in New York with a histone marker and a membrane marker. The idea was here to do lineage tracing in C. elegans. And you hopefully see that imaging, very fast imaging you can do with the system over a decent period of time, and you can see the green signal emerging over time, which basically was the idea. In another application example, we imaged a large 2D or 2.5D 3D cell culture of human primary cells. This is now 300 by 200 by 1200 micrometers, so it was really a very large tissue section provided by the Tucker lab in Cambridge. And there we can also obtain really very nice 3D overview images, but at the same time, very high subcellular resolution, as highlighted in this movie. When zooming into this large data set, you will see that when looking at individual nuclei, you can really see the subcellular and the subnuclear structure of the chromatin here, for example. So the system provides a very large field of view, but at the same time, a very high subcellular spatial resolution, and all the advantages of light sheet microscopy. I mentioned already that we offer two sets of lenses. One would be the Nikon lenses for live imaging, and the other one, the ASI special optics lenses for also imaging of cleared samples. And that's an example of the mouse lymph node that was cleared with BAP, imaged on the QV light sheet microscope. So the application areas of this QV SPIM are quite versatile. It ranges from really cell biology applications up to high content imaging, because you can really image many samples that you can distribute over an 80 by 120 millimeter SBS plate, but you can also image cleared samples. So, which takes me to the last point, light sheet microscopy of cleared samples on Luxendo light sheet microscopes. Why do you want to do cleared sample imaging? As you know, if you want to image a whole embryo or the brain of a mouse, for example, the objects are just not transparent. They scatter a lot, they absorb a lot. So if you clear samples, and this is very, very dynamic, it's a very dynamic field at the moment, you basically try to reduce or eliminate the absorbing and the scattering objects in the tissue, which is very often lipids for the scattering. And there are just a really broad range of protocols and procedures how you can do this, so that such a large object of up to 10 or 20 millimeters across in diameter become transparent enough so that we can image this in a fluorescence microscope. And also all these protocols have to make sure that the fluorescence labeling, either with immunofluorescence or with fluorescent proteins, is not affected. So there's really, typically you go several steps to remove this, and I don't want to go into detail on all the clearing approaches. The methods can be grouped into solvent-based methods, water-based methods. Some are just based on simple immersion and diffusion. Others require hydrogel embedding. I don't want to go into the details, but the concept is you remove scattering and absorbing molecules so that a large object becomes transparent enough to be properly imaged on a fluorescence microscope. The challenges of imaging cleared samples, especially on a light sheet microscope, are sample mounting. The sample is quite large, typically in the range of a centimeter or larger, and we have to be able to typically position the sample in front of the objective lenses. Then the objects very often deviate within their refractive index from the solution you're using and also from water, so we have to somehow have a tool to correct for refractive index mismatches. And since the objects are quite large, we need objective lenses that have a working distance that is large enough and long enough to image these objects of several millimeters or even centimeters in size. So we try to address all these challenges with an add-on to our MovieSPIM, which we call the MovieSPIM CS. It's a cleared sample module, which, as you can see, is just an alternative octagon sitting on top of our light sheet microscope, and we also then mount the sample from above with a stage instead of mounting from below, as we do this for the previously introduced live sample MovieSPIM. So as I said, we address all these issues in a way that we use Nikon objective lenses, either a 20x1.0 and a multi-immersion lens with a working distance of 8 millimeters, covering a refractive index range from 1.44 to 1.50, or a Nikon 10x.5 NA lens with a working distance of 5.5 millimeters at a refractive index range of 1.33 to 1.51. So while these two lenses cover a very broad range of refractive indices, they still have a very high resolution and they have a very good chromatic and chromatic correction. So these are really great objective lenses. For illumination, we take air lenses, so they don't have to dip into the chamber. And we usually suggest to do single-sided illumination to be able to image also larger objects. So looks like sharing is paused at the moment, so give me a second to restart the presentation. Apologies for that. Also, with these two objective lenses, we can cover a quite broad range of fields of use and pixel sizes, really depending on your needs, whether you need more an overview image or really high resolution. And our system is really very well suited for high resolution imaging of cleared objects. The two objective lenses provide a correction collar for refractive index correction, which then allows to remove the aberrations introduced by the variations in refractive index of your sample. So the sample sizes we can look at are up to 12 millimeters laterally or in one direction, and up to 60 millimeters in the other direction, and up to, let's say, 15 to 90 millimeters in the vertical direction. And with the rotation stage that we have implemented, we can then image from two sides so that we can image the whole objects up to 60 millimeters in depth. We are using Nikon lenses, and Nikon provides a very nice table of compatible clearing reagents. And it turns out that most of the clearing reagents are compatible with the objective lenses. Most of them are compatible in terms of corrosion, and not all of them are compatible with the refractive index range. But what we have learned using the system is that even outside the nominal specifications of the objective lens, so beyond 1.5, for example, when using, for example, one of the disco reagents, we can still image very nicely objects, even though this is nominally outside the spec. So I would say that even with 1.56 refractive index, we can generate very nice images, as I will show you. Sample mounting is from above, as said, and we are very flexible there. We provide basically an interface for a flexible geometry of sample holders. We offer 3D printed sample holders. We offer also files so that you can design your own sample holders. This is based on this. You can just put your sample, like this little mouse brain here, on this little platform. We also support hook mounting, skewer mounting, hanging, or cuvette mounting. This is another example of objects that were then basically squeezed in between. How do we switch between the live sample and the cleared sample configuration? That's very easy. As you could hopefully see, you basically just remove the live sample octagon, take it off, and then put on the cleared sample octagon, and within 10 minutes, the system is ready to go with another, for cleared samples rather than live samples. In addition, you can also remove we can remove the central part just containing the sample, so that you can just take out the sample and take the cleaning of the sample chamber offline and just put another sample in the meantime, so that there's no downtime of the system. Especially in multi-user environments like a facility, this is hopefully very interesting because it's very easy to swap between live and cleared sample imaging. It doesn't take much downtime. It doesn't result in much downtime, 10 minutes. And you can also very easily switch between different samples so that there's no delay between imaging, especially when you're imaging large objects. This takes a while, and then you don't want to lose time to optimize your throughput. Of course, the system supports tiled acquisition, which then also, and our software also provides the functionality to stitch these different tiles together. And this is an example, not the most obvious one. That's an octopus provided by Montse Collado from EMBL in Barcelona in Spain, which has been cleared with TDE, a very nice example. That is a cubic cleared brain, also provided by EMBL Barcelona, in this case Jim Swoger, where they could image really over a very large field of view and also generate nicely depth color-coded views on the object in this part of the field of view, for example. So the range of applications is as broad as I mean, you can image basically everything you can clear with all kinds of reagents, because we cover this broad range of refractive indices. That's an example of a BAP cleared mouse head. Here you can see that the refractive index is nominally outside the range, but the imaging worked very well because we can, with our very flexible illumination optics, we can very easily focus the light sheet again and bring it into the right focal plane of the objective lens. This is also a tiled and stitched acquisition. And this is the lymph node that I showed you already imaged on the QV SPIM, and this is just the same sample imaged on the movie SPIM as well. Okay, and that's another example of a cleared mouse brain slice, approximately 500 micrometers thick, in a couple of millimeters in lateral dimensions. And you can hopefully see that while you can get nice overview images, you can also get a very high resolution when zooming in to the object. And that's another example of a cleared mouse brain from a customer in China, where we see very nicely individual neurons that can be resolved even very deep inside the tissue. Okay, as said, that the upper limit of sample sizes for imaging of cleared samples in the movie SPIM is in the range of, let's say, 10, 12 millimeters. But what if your samples are larger? If you need to image larger objects that are beyond that? We thought about that, and then we said, okay, we can make use of our flexible approach again, and we follow the same geometry approach as we did for the true live 3D imager. We are taking an inverted geometry to image from below and to illuminate from the sides. Then using just air lenses, so dry immersion lenses for illumination and for imaging, we have a longer working distance available, and then we can image really larger objects embedded inside cuvettes. Everything else is the same, so we are using CMOS, high sensitivity CMOS cameras for detection of 1 or 2 imaging channels. We can offer up to 6 or 7 laser lines, ranging from 400 to 800 nanometers, and the samples are mounted in a standard quartz crystal cuvette, so that you can very easily swap from one sample to the other without having to clean objective lenses. So this is really a benchtop system that is optimized for high throughput. That is a visualization of the optical concept. So we're basically illuminating from 2 sides with 2 4x.2 NA objective lenses, and instead of moving the sample to acquire to laterally acquire the whole object, we are moving the objective lens so that we minimize the need for movement, so that we also minimize the motion-induced distortion of the sample. In Z, we still have to image, we still have to move the object to acquire a full 3D stack. What we also make use of is fast axial scanning, so that we do not only have a the thin part of the light sheet in the center, but across the whole field of view, so that we can image basically with a defined light sheet thickness across the whole field of view and across the whole quartz cuvette. This is how you basically put on the cuvette. So it's very straightforward. Then you close the lid of the microscope and you can start imaging. This summarizes this. So the refractive index range goes from 1.33 to 1.57 that we can image with this. And the mounting is really straightforward because it's just imaging inside a cuvette with sizes between 20 by 20, 20 by 30, and 20 by 40 millimeters. And there's more to come. With this, I would like to come to the end. I walked you through our product portfolio of different light sheet microscopes based on different geometries and inspired and motivated by basically the needs of the different kinds of samples. And that's basically the idea. It depends on the sample, what you need, and we are trying to offer you a solution for your application and for your sample. This is our product portfolio. I talked about the movie SPIM in its different implementations, the live sample version, the photomanipulation module, the cleared sample version, and the combination of both, the InVSPIM, the inverted light sheet microscope, that can be complemented by the advanced illumination module for more complex illumination with bezel or lattice beams. We have our upright model, the QVSPIM, for very versatile imaging ranging from cell biology to cleared samples. We have our large cleared sample SPIM system for imaging of very large cleared samples, and especially for applications in the field of organoid and spheroid research, we have a dual-sided illumination July 3D imager. This is complemented by powerful acquisition and processing software, and we also offer a solution for storage and processing to help you managing your data, and the amount of data you generate with light sheet microscopy is usually quite large. With this, I would like to thank you for your attention. I'm very happy that you joined, and should you have more questions, don't hesitate to contact us through our website, for example, and we are happy to move on with any discussions. With this, I would like to finish. Thank you very much, and stay safe and healthy.